So without further ado, I'm going to say welcome everyone to the Tennessee Native Plant Society's seminar on Fungus Among Us, an introduction to fungi for the home gardener by Alisha Millican, who is a new member of TNPS and she recently moved from Alabama to Tennessee. And she really knows her fungi. She's going to give us, I'm sure, a lot of good tips and lots of information. And I'm sure answer a lot of our questions. So I could go on and on. You've got a lot of good credentials here. And she's active with actually, she's current president, soon to be retiring president from the Alabama Mushroom Society. You might see their logo on your screen somewhere there. And she serves on the executive board of the North American Mycological Association and is assistant curator of fungi for the University of West Alabama Herbarium. She's a dedicated advocate for promoting fungal education and stewardship. Alisha enjoys exploring nature with her husband and 2 children when she's not immersed in mushroom hunting or gardening pursuits. You will probably enjoy going on some of our field trips. And depending on the kids' ages, you might want to bring them along. So
they're 2 and 4, so
not
maybe
yet.
here in another couple of years. Yeah.
Not yet. Down the road a little. Okay.
Yeah.
Without further ado, folks, let's welcome Alisha Millican and our talk on fungi. So without further ado, I'll let you take it away, Alisha. And I hope I'm saying your name correctly.
It's Alisha, but I don't take any offense. It's
okay.
all right. So, Karen did such a great job of introducing me. We're going to touch on fungi as a kingdom and kind of a very brief science lesson as to what are fungi and why they're different from plants. And then bear with me, it does get a little bit technical there, but we're going to back off a little bit. And if you have any questions, please type them into the chat and we can address them at the end. Karen did a great job of listing off all my credentials there. I was a master gardener in Georgia and then Alabama, so plants were my first love. And I kind of serendipitously wound up at an Alabama Mushroom Society meeting in 2020 and got sucked into the world of mushrooms and was elected the VP of AMS in 2021 and then assumed the role of president. And then very quickly became involved with the North American Mycological Association and then took on the Southeast Rare Fungi Challenge with the Fungal Diversity Survey in 2022. And then last year took on my assistant curatorship of fungi at the University of West Alabama. And am happy to be on board as a member of Tennessee Native Plant Society. So we're going to start off with some of the basics. What are fungi? You may remember back to your days of high school biology that all life is categorically placed into increasingly smaller groups through the science of taxonomy. These groups, from largest to smallest, are kingdom, phylum, class, order, family, genus, and species. As an example, we're going to review the taxonomy of a domestic apple. Oh, sorry. Got to remember that I'm clicking on my desktop and not my laptop. All right. So apples are in kingdom Plantae, phylum Magnoliophyta, class Magnoliopsida, order Rosaliae, family Rosaceae, genus Malus, species Domestica, and all domestically bred apples are varieties of the same species. All life falls into one of 3 to 7 kingdoms, depending on when you were in high school and which taxonomist you ask. Fungi are the organisms that fall into the kingdom Funga. Kingdom Funga is closer related to kingdom Animalia, which includes us, than they are to the kingdom of plants. However, up until 1970, 1968, they were placed as plants. They were considered some of the cryptogams with ferns and mosses. So it's fairly recently that they have been recognized as an entirely different kingdom. Kingdom funga includes yeasts that rise breads and brews beer, molds that decay foods or make blue cheese, human pathogenic fungi like thrush and athlete's foot, rusts that cause plant diseases, and mushrooms. Let's talk about that word, mushroom. Mushroom is defined as the fleshy, spore-bearing fruiting body of a fungus, commonly thought of as the standard cap and stem shape of your standard grocery store varieties, but they can come in many other morphologies, like cups, pits and ridges, corals, and tentacles. They can be little bitty, and they can be absolutely giant. And this is a real photo of a real fungus that actually occurs in Florida and Alabama as well. This is called Microcybe titans. This mushroom here is Agaricus bisporus. This is the mushroom that you will find in the grocery store or in your Campbell's cream of mushroom soup. When you see mushrooms listed as an ingredient on a food label, this is, per the FDA, the species that is being referred to. It goes by the common names of cremini, portabella, portobello, white button mushroom, and brown button mushroom. These are all the exact same species. They are just at different stages of maturity. This is the only mushroom that many people have ever eaten. So I'm going to give you a bit of a comparison. I want you to think about the differences and of flavor and texture between eating a mango and the flavor and texture of a pine cone. These two plants are as closely related as this button mushroom is to some other edible mushrooms, like our morels. So if you never ate another plant because you once ate a pine cone and didn't like it, look at all you would be missing out on. There are mushrooms that taste sweet, spicy, bitter, fruity, earthy. There are some that taste like licorice, maple, bleach, anise, and many other flavors. To say that you don't like mushrooms is writing off a very wide variety of tastes and textures, usually based off of a single mushroom that is only widely eaten because it is easy to cultivate and it ships well. So before we go any further, I want to address some mushroom myths. And at the end of this, if you have any myths that you've heard that we can discuss, be thinking about those. The first one we're going to talk about is if the birds or the bugs or the animals are eating it, it's good to eat. This is absolutely false. Animals can and do eat and die from toxic mushrooms. They also have very different body systems than ours and have different reactions to toxins than we do. You can think about chocolate, which is toxic to dogs and is safe and delicious to humans. Use a mesh bag or a basket to gather your mushrooms to spread the spores, or tap on your mushrooms to spread the spores. You're welcome to do these things if it makes you feel good, but mushrooms drop billions to trillions of spores every day. Any mature mushrooms that are ready to be collected to eat have already been dropping spores, and the comparatively few that you may stimulate them to drop probably isn't making a considerable difference in any future yield. It does, however, demonstrate a generous spirit. I wouldn't discourage it, but it's not necessary. Poisonous mushrooms will turn a silver spoon or a coin black if you boil them with the spoon or the coin. This is false. There's no known mycotoxin that is known to turn silver black. This is just baseless folklore. Mushrooms are dangerous to touch. This is something that in North American culture, many of us have been raised to, be afraid of mushrooms. Don't touch them. Go wash your hands. They have a lot of negative connotations, but this is also completely false. Even the most toxic mushrooms are completely safe to touch. There is no known mycotoxin that causes any sort of topical reaction. If you do have a topical reaction to a mushroom, it is not necessarily indicating the toxicity. It may just be an individual reaction, which leads us to our next myth. If you rub it on your skin and if it's toxic, you will have a reaction. This is crossover advice from old school plant edibility tests that do not apply to fungi and aren't great for plants either. But I actually have an old Army manual that gives these instructions. It wasn't good then, it's not good advice now. And we have the famous cut, don't pick. This is a hotly debated topic. From the very few long-term studies that have been done, we know that cutting versus picking makes no significant difference in the future yield of the one type of mushroom that has been primarily studied. But applying those findings to every single fungi without further research is kind of ludicrous. More research is needed. That being said, mushrooms do not have roots like plants. They have mycelium, and the mushroom is the fruiting body of the mycelium. For the grand majority of the common edible species, it behaves like the apples on a tree. Picking the apple does not hurt the tree. Do not pick more than X% of a patch of mushrooms. 20% is the number that is commonly thrown around. This is more crossover advice from the world of plants. It would be ridiculous to suggest that picking 100% of the fruiting bodies from a patch of mushrooms wouldn't impact it, but it is not realistic or practical to do so. There are always some that are too old, too damaged, too young, or otherwise not suitable for your basket. Sexual reproduction. Sorry, I've lost my place. Sexual reproduction from spores isn't the only way that mushrooms reproduce, and the underground mycelium, which is the actual fungus, will put up more fruits even if there are no fruiting bodies dropping any more spores. Does anybody have any mushroom myths that you've heard of that you'd like to mention?
I don't see any new ones
If
in
anyone
the
thinks of
chat.
any, drop them in the chat and we will touch on them at the end.
I have one, Elise. My name's Hill.
Hi, Hill.
If once you see the mushroom, they stop growing.
No,
Which
that's
you might as well pick them all, even if they're tiny, because they stop growing.
that is, I can definitely attest from personal experience from studying mushrooms for years and going back to photograph them and document their growth that is not true. I've also heard that morels pop out of the ground fully formed and that they make an audible popping sound. That's a fun one. That is also utterly ridiculous, but I've heard repeated many times. We got one in the chat. Blue stainers cannot be eaten. Negative. There are lots of different reasons that mushrooms stain blue, and I can list, gosh, 24 bolete specimens that species that stain blue, that it's just general oxidization and they are perfectly safe to eat. So that alone is not a shortcut to knowing edibility. Yeah, yeah. People are yes. People, the louder someone is about mushroom facts, the more wrong they probably are. I think I saw somebody with their hand up.
Mm,
Did we miss
I-
anyone?
I don't see any.
Okay. All right. If anyone thinks of any others, feel free to drop them in the chat and we'll discuss them. Okay, so I do want to touch on a little bit of foraging etiquette, and this comes from the book Wild Mushrooms by Kristen and Trent Blizzard. Trent Blizzard is the current president of the North American Mycological Association. And these are the things that actually matter, unlike many of the things that we just listed. Don't trample an area. Trampling damages the mycelium. It can inhibit mushrooms from fruiting. It's also dangerous and detrimental to the plants. And trampling plants doesn't just impact that plant, it's bad for the mushrooms. More than 95% of terrestrial plant species form a symbiotic relationship with fungi. And mycorrhizal fungi, they connect to the plant roots in the soil and act as an extension of the plant's root system. They send nutrients and water to the plant, often in exchange for carbon and sugars. And so by trampling and killing the plant partners, you're removing a nutrient resource from the fungi. Leave no trace. Not littering goes without saying. In fact, if you could take a trash bag with you and pack out some garbage that somebody else carelessly left behind, would be advisable. Beyond that, travel as invisibly through the forest as possible and leave some for others. Consider leaving some fungi behind for each patch for other foragers, for hungry animals and insects, and because it demonstrates a generous spirit. So where and how can you go about learning to forage for mushrooms? Hands-on experience is best. There are important aspects of mushroom ID that cannot be conveyed from photos in a book. The smell of an anosogby or the texture of a rustula can only be learned with the mushroom in your hands. So where do you find that hands-on experience? Your local mushroom club, of course. The North American Mycological Society has a list of affiliate clubs on their website. The only official club in Tennessee is the Cumberland Mycological Society, which is up near Knoxville. But we are in this beginning stages of starting one in the Chattanooga area. I highly recommend doing extensive research into the qualifications of anyone that you are paying for a private class. There are a plethora of survivalists and herbalist folks educating on mushrooms right now because it is the hot, popular topic and they have no business doing so. I've corrected and seen some pretty awful advice getting thrown around. There are excellent free online mycology lectures available on YouTube or from many different mycology clubs, including the Alabama Mushroom Society. We hold a free public presentation from an expert every month via Zoom. The Central Texas Mycological Society frequently holds online lectures, and NAMA often has lectures that you can pay to attend. Feral Foraging and Learn Your Land are great YouTube channels to learn about some wild edibles, including mushrooms. And never, I seem to have my notes in the wrong order here. Never eat a mushroom unless you are confident in its ID. Getting online ID suggestions should be a starting point to your own research to confirm that suggested ID and that it matches the mushroom that you have in your hand. I'm not sure what has happened here. I've skipped some online platforms, I think because I'm going to touch on those here in just a second. Know that the grand majority of mushrooms have to be cooked to be safe to eat. Even delicious edible species can cause nasty GI upset when raw. Get a current and up-to-date field guide. Mycology is a rapidly developing science, and field guides from the 80s and 90s have just been so quickly outdated. Some of the best field guides that are applicable to Tennessee include Mushrooms of the Gulf Coast States. This is probably my top choice here by Alan Bissett, Arlene Bissett, and David Lewis. Mushrooms of the Southeast by Todd Elliott and Stephen Stevenson is a decent guide to some of our more common species. And then if your primary interest is foraging for edible species, your must-have book is 100 Edible Mushrooms by Michael Kuo. It's a fantastic beginner's book. The first couple of chapters walk you through everything you need to know as a beginner and how to keep you safe while trying wild fungi. And the chapters are sorted by ease of identification, and it includes full color photographs, preparation instructions, and even recipes, which I think is a nice touch. You may note that Tennessee is not listed on any of these books, but they are absolutely applicable to this area. And also, please be aware that any mushroom field guide that you find online right now, specifically for Tennessee, is written by AI. They contain straight up misinformation. Many of them are unreliable and dangerous. There is no published book for mushrooms of Tennessee by a mycologist available right now, but you will find them on Amazon. They are no good. And then there are a variety of online platforms. We're going to talk about them here. So a couple of resources. First are our Facebook groups. The first one I want you all to note down. This is Poisons Help Emergency Identification of Mushrooms and Plants. It's a group made up of hundreds of plant and mushroom experts from around the world. It is for emergency ID if you have come into contact with an unknown plant or eaten an unknown plant or confirmed ingestion of a mushroom has occurred. It saves lives. Doctors at emergency rooms cannot ID. They cannot do their job. Even toxicologists cannot do their job without a positive confirmation of a plant or a mushroom. For less emergent situations, us over at the Alabama Mushroom Society welcome posts from across the Southeast, as well as mushroom identification and the mushroom identification page. If you aren't on Facebook, some good website resources are the page for the North American Mycological Association, namyco.org, the Alabama Mushroom Society.org, and two sites where you can post photos of mushrooms and get suggested ID by experts include iNaturalist.org and mushroomobserver.org. Both these sites log important data about what species occur where, and they help mycologists know species ranges as well as rarity and what species may need to be protected. I am specifically on these sites IDing, and I tend to ID where I'm at. So if you're in Tennessee, especially if you're on the eastern part of the state, I do a pretty good job of trying to throw IDs on all the mushrooms that I can. So you found a mushroom you want identified. Now what? These are the things that you should be making note of and the photos that are needed to get an ID. You want a photo of the mushroom undisturbed in its habitat. I need to be able to see if it's growing from the ground or on a log, if it's clustered in a group or growing alone. And then you need to dig the mushroom completely up without cutting or breaking it. I need to see the complete base. Some mushrooms have deep rooting stipes, while others have a cup called a vulva around the base. Take an up close and an in-focus photo of the cap, the mushroom taking up the majority of the photo. I don't want to see a mushroom from across your yard and have you say, oh, hey, Alicia, tell me what this mushroom in my yard is when it's a tiny white dot on your screen. I'm not going to be able to help you very much. Flip it over and take a photo of the underside showing if it has gills or pores or teeth, a photo of the stem. Often cut long ways from top to bottom can be very helpful, taking note of any color change. For example, if it's turning blue, like this bolete shown in this photo here. Take note of what trees are growing nearby. If it's growing from wood, is it hardwood or a conifer? Is the wood living or dead? If you crush a little bit of the cap and smell it, what does it smell like? Some examples of officially documented mushroom odors include old gym socks, drying ham, almonds, lemon, maple syrup, hyacinth, cinnamon, cocoa, green corn, potato, coal tar, and chlorine. And I'm telling you guys, some of these mushrooms will be in my box. And by the time I get them home and I open that box, the smell of the coal tar or the chlorine just about knocks you over. I mean, these are sometimes not subtle odors, and people don't think about mushrooms smelling strongly, but I can tell you there are some that do. So when you consider that expert identifiers need all these photos, all this information to help identify your mushroom, you may be wondering how apps on your phone can do it with just one photo. And it's because they can't. Apps are dangerously unreliable and full of really bad IDs and misinformation. Even professional mycologists cannot ID many mushrooms to species without microscopy and sometimes even DNA sequencing. The app technology just is not there yet. Do not waste your time. The closest thing is the AI suggestions on iNaturalist, which I was at a 4A this past weekend, and iNaturalist was darn convinced that my mushroom was a banana slug. So they definitely have their limitations. All right, so some of the good stuff. We're going to talk about some of the common edible mushrooms that you can find right here in Tennessee. This is meant to be a brief introduction. It's not meant to be comprehensive, and it's certainly not intended to be a thorough enough walkthrough for you to go out and ID a mushroom with enough accuracy to take it home and eat it. It is meant to whet your appetite and spark your interest to hopefully learn more. Puffballs. They are a form group that include the genera Lycoperdon, Apioperdon, and Calvaceae, and some smaller genuses that we're not going to talk about. They don't have a standard cap and stem structure. They range in size from the size of about a quarter to the size of a basketball, and they're just kind of oblong balls. You'll find them commonly growing in grass in the summer and fall, oftentimes on your lawn. One species, Apioperdon periform, which is the pear-shaped puffball, grows on dead wood. There are a few toxic lookalikes that you can rule out by texture. If it cannot be squished like a marshmallow, it is not a puffball. These, you squish them, they squish right down. If you squish it and it just shatters to pieces, it is not a puffball. Amanita buttons are a toxic lookalike, and in addition to being much firmer than a marshmallow, if you cut one in half from the top to bottom, they will have the outline of a mushroom inside, which is what you can see in the middle top here. The one pictured is orange and yellow, but the outline can be all white. If the inside of the puffball is anything other than pure white and the texture of a foam mattress, do not eat it. If it is very firm or starting to turn yellow, green, or brown, cast it aside. It's too far gone in its maturity. These have no flavor on their own, but they take up the flavor of whatever you cook them in. You can slice them into steaks and grill them. I consider them kind of meh in desirability. They sort of have a tofu-like texture, but they're edible and super easy to ID and commonly found in your lawn. Lobster mushroom, Hypomyces lactiflorum. It's a mushroom parasite, actually, that takes over these boring russulas, making them very tasty. This is a Russula breviipes before being parasitized, right here on the right. And that's that same mushroom after being parasitized. You can see that it completely takes over the gills, giving it kind of a lobster claw look. You'll find these in the woods in the fall. You want to collect the ones that are very firm, like a raw potato, and discard any that are smelly, soft, or soggy. My favorite way to eat these is to grate them up and to make them into latkes. Chanterelles, the Cantharellus species. There are 23 species of chanterelles known from the eastern US and an estimated 15 to 20 that are still undescribed. But the color ranges from red, orange, and yellow to pink and buff. They're popping up everywhere right now. Some of the defining characteristics of chanterelles are they do not have gills like the pages of a book. The spore-bearing surface is made up of veins, ridges, and wrinkles with cross-veining going between them. When torn from top to bottom, the mushroom should peel like string cheese and be white inside. Look for these in areas of hardwoods all summer and fall, pretty much till we get a freeze, and so long as we're getting plenty of rain. Big species of chanterelles are delicious added to pasta, and the teeny tiny cinnabar red chanterelles I enjoy candied and served over vanilla ice cream, which is not a way I bet most of you have thought about trying mushrooms, but they are delicious. Craterellus species are the black trumpets. The two most common species in Tennessee are Craterellus phalanx and Craterellus foetius. They are black or gray, often with a yellow cast to them. They grow in association with hardwoods in areas where moss grows, and they're found in the summer and fall. They're trumpet-shaped and hollow all the way down. Their most convincing lookalike is Urnula craterium, the devil's urn, which always grows up against dead wood and occurs earlier on in the spring. It's considered inedible, but it's not toxic. They have a very black trumpets have an earthy taste to them, and I enjoy them added to Alfredo. Chlorotus species are the oyster mushrooms. They can be found year-round on dead hardwoods. They're best collected during the fall and winter because spring and summer oysters tend to be really buggy. Our native oysters are white and cream to a gray-brown on the cap, but there are cultivated species that are blue and pink, and there's an invasive species that is bright lemon yellow. Their defining characteristics are an anise or fishy odor and having gills that run down the stem. They're very mild in flavor and they're firm, so they make a great dexel or an addition to stuffing. We have two species of cauliflower mushrooms, Spiraceus americana and Spiraceus spathulata. They look like somebody went out and dumped a pot of noodles on the ground in the woods. They grow from the roots of pine and oak, but will look like they are growing from the ground. They only come up once a year and they form a hyphal mass called a sclerotium. So they should be cut at soil level to avoid stunting them. It's one of the few mushrooms that I would say cut versus pick has kind of a clear answer. The closest lookalike is not toxic and will have small spines or warts on the flat underside. I'm sure it's been eaten dozens and dozens of times by mistake. It's non-toxic. It's a little bit tougher. Spiraceus will be completely smooth on both sides of the fronds, and their firm texture makes them a really great substitute for noodles in soups or other really wet dishes. Ericium arenaceus is lion's mane, typically found during the fall and winter on standing or propped dead hardwoods. They are a tight ball that grow teeth 1 to 5 centimeters long. There are quite a few of similar looking fungi that have spines but are not in a tight ball, or mushrooms that are a tight white ball but do not form spines, none of which are toxic. It has to be both a tight ball and have the spines to be a lion's mane. They have a texture very much like crab meat and make delicious mock crab dip or mock crab cakes. This is one that I always say I'm going to do something else with when I find it, and I always make mock crab cakes with it, and it's just really excellent. Laetoporus species. This is your chicken of the woods. We have two species here in Tennessee, one with a white underside and one with a sulfur yellow underside. They all have white inside flesh if you cut it open. The closest lookalike is Pseudophaeolus trichoris, which grows on Quercus virginiana, and it has peach-colored flesh inside. It's non-toxic, but it's very quirky and tough. Laetoporus has a higher than average report of folks having individualized sensitivities resulting in GI distress. So we recommend anyone trying this one for the first time only eat a couple tablespoons and then waiting 24 hours to see how your guts handle it. It's good advice when trying any new food, but especially pertinent for this species specifically. And like most mushrooms, it should be cooked really well before consuming. They're mild tasting and have a texture just like chicken. It makes a very convincing chicken or meat substitute in any dish. These are the wood ears, Auricularia. We have at least 5 species of wood ears. They're all pretty similar in appearance with varying degrees of fuzziness on the outside. They grow on the dead wood of both conifers and hardwoods and are pretty commonly found year-round. They have a firm jelly-like texture and form individual ears that may be fused but do not form an indiscernible clump. There are several jelly fungi that can be confused for auricularia, but they're all edible. These are commonly eaten in Asian countries and are traditionally served in hot and sour soup. I think they have the texture of like chicken gristle, and I don't like them. All right, I am going to address one question that came up just before we dive into the non-edible fungi. I was asked, the question was, oh, I think I skipped it. What is the difference between chicken of the woods and hen of the woods? Totally different species. Yeah, hen of the woods is Griffola frondosa. It's brown, it's a fall mushroom. It's just it's similar name. We have lots of fowl of the woods, bird names for fungi for whatever reason. It's completely unrelated. Also edible. Oops. Okay, fungi in the garden. We're going to start with the good guys. Hohenbuehelia petaloides. Guys, I'm gonna quiz you at the end, so be memorizing these. I'm just kidding. These are the shoehorn oyster. They grow on woody debris. These are a fantastic find in your garden. They grow on mulch or buried wood, and the mycelium in the ground that sprouts these mushrooms, it's made up of cells called hyphae, and the hyphae of Hohembohalia petaloides are carnivorous. They trap and digest microscopic nematodes in the soil, cause a variety of plant diseases. They're technically edible, but they're so woody and tough that I don't know why anyone would bother with them. But yeah, these are, you can see a very high-powered microscopy of this. I think this is like an electron microscope of the hyphae forming loops. There are many carnivorous fungi, and they kind of trap nematodes in different ways, and these form little nooses that strangle the nematodes, and then they digest them. So you want these in your garden. This is the Nidularaceae. They are a family of fungi known as the bird's nest fungi, and for good reasons. They look like tiny little bird's nests with little eggs inside. They're very, very small, from about 0.5 millimeter to 8 millimeters at largest. They grow on wood chips, and soil containing woody debris. Absolutely harmless. They're just breaking wood down into nutrients that your plants can use. They're considered inedible because of their teeny tiny size, but they're an absolute delight to find. They come in different colors and textures, and you got to kind of get down there with your magnifying glass, but they are just gorgeous little fungi that you might find on in your mulch beds. Leucocoprinus species are a common find in container plants, both indoor and out. They typically pop up in well-watered potting soil. It's not a problem in hardy plants, but it should cause you a bit of concern if you have these popping up in your succulents, because they're too wet and you may be risking root rot. The mushrooms themselves, harmless to your plant. They're mildly toxic if you were to take a big bite of them. They range from white here on the left, these are different species within this genus, to various shades of yellow, but they all kind of fall in those same categories. Mildly toxic, harmless to your plant. Enjoy them while they're there. They won't last long. The Phalaceae are the stinkhorns, and they are aptly named. You will often smell these before you see them. They all have a stinky spore mass called gleba that attract flies to spread their spores. They start out as eggs that emerge from the soil and are technically edible in egg stage, but I don't think I've ever been that hungry. Like all mushrooms, these are safe to touch, but you will wish you hadn't, because the smell may cling to your hands for days. These will even look like little alien eggs here with their tentacles sprouting out, but again, completely harmless, a little bit stinky, no concerns. Now we're going to talk about some of the not so good guys. Mycosarcoma matus, this is corn smut. If you've grown corn, you will likely recognize this fungus. It's known as huitlacoche in Mexico, where it's commonly eaten. It's a delicious edible. It doesn't have any lookalikes. You just remove the firm gray baubles from the corn and saute them up in some butter. It has a very distinct taste of both corn and mushrooms. I absolutely love it fresh in tacos and quesadillas. However, it's on my bad guys list because it's not a great omen for your corn. Diplocarpon rosae is the black spot of roses. It affects all species of rose. It sporulates when it is between 60 and 80 degrees and wet, but will not spread if the leaves dry out within 7 hours. The spores are spread by wind, rain, and handling, which is why you should not handle your plants when they are wet, as this is how many pathogens are spread. If left untreated, it can cause defoliation. It can spread to the stem of the rose and will cause progressive decline of the plant. It will overwinter on infected material, so infected material should be removed and disposed of by burning. Management options include proper pruning and spacing to increase airflow, which will prevent spreading and infection. You can apply fungicides labeled for black spot at the very first sign of infection. You can grow black spot resistant varieties. There's a product called Serenade that contains the bacteria Bacillus subtilis. It can be used preventatively and is safe for our pollinators, and is actually an effective prevention for many fungal diseases of plants. Cochineales, these are our rusts. These are the fungal pathogens that cause diseases in plants. They have a complicated life cycle that can include up to 5 types of spores that are each born on a different type of structure and take place between their primary and secondary hosts, and sometimes even include a tertiary host. Having multiple hosts with different spore-bearing structures means they are very difficult to treat. Fungicides may help, but they never truly eradicate the disease. Some organic preventative solutions are available, including sulfur powder and products containing Bacillus subtilis. High standards of hygiene, good soil drainage, and careful watering can help minimize problems. Any appearance of rust must be dealt with by removing and burning the infected debris. Composting or leaving infected debris on the ground will spread the disease. There are an estimated 168 genera within the family, which is known to contain 7,000 species, which is guaranteed a gross underestimation. This is a really wide and complicated group of fungi. So one of the rusts that you may be familiar with is Gymnosporangium juniperi virginianae. It's the cedar apple rust. Its primary host is apple and crabapple trees, and its secondary host is the eastern red cedar, aka the juniper. On the left here, you see the spots caused by this fungus on an apple leaf, and on the right is the mature gall on a juniper. The life cycle starts on an apple tree when the spores land on the leaves and show visible infection within 2 weeks. It forms clustered cups on the bottom of the leaf, which drop spores in June and July, which can be carried up to a mile to infect junipers. At the end of the second year, small galls will form on twigs of the juniper. The galls mature and form bright orange horns in the spring and will discharge spores 3 to 5 hours after the first warm rain of the year. These spores can travel up to a mile to infect apple trees. So you can see why this life cycle that includes all the apple trees and juniper trees within a mile of any of the infected trees can make treatment very complicated and difficult. The top photo is the gall on a juniper, and the bottom is showing the development of the spore horns off the gall. They do not damage or cause any harm to the juniper tree. Management options include planting resistant apple varieties, cutting down all junipers within 1 mile of your apple trees. There was actually a Supreme Court case in Virginia that allowed for the removal of all eastern red cedars within 2 miles of any apple orchard if an official investigation found the cedars to be the host of this disease. Pruning away and burning all the galls from any junipers within a mile of your apple trees. Spray either or both trees with fungicide. By the time the apple trees are showing spots, it's already too late to treat them. There are very similar species that also infect pear, quince, hawthorn, and other trees. Phrena deformans is the fungal pathogen that causes peach leaf curl. It's extremely common and present in most peach orchards worldwide. It affects trees within the Prunus genus, including almonds, peaches, apricots, and nectarines. Interestingly, the strain that affects almond trees will not spread to peach trees and vice versa, so future studies may separate them into separate species. Infection will present with a white yeast-like bloom on the leaves, which will progress to curling, purple lesions, pale yellow, green discoloration, defoliation, deformed fruit, fruit drop, and can even kill young trees. For home gardeners, there are resistant varieties of peach, but these are not suitable for commercial production. Even varieties with relatively good curl resistance are somewhat susceptible during the first few years of growth. T. deformans loves years with warm, wet winters or cool, wet springs. It needs at least a ½ inch of rainfall, followed by at least 12 days of temperatures between 46 and 53 degrees in order to fruit and spread. Treatment after infection is inadequate to control the disease, so prevention is your key. Applying both a fall and spring application of fungicide is recommended in wet climates. Chlorothalonil and Xyram are favored, and copper is an organic option. So I get asked this all the time. Many people want to know, how do I keep mushrooms from growing in my yard? If you type that question into Google, you will find dozens of articles written by lawn care companies and bloggers full of wildly misID'd mushrooms and advice such as covering the mushrooms in dish soap, salt, or baking soda, spraying commercial copper solutions, or even removing all the soil from your yard. So which, if any, of these solutions have any merit? Before we talk removal, I want to express that the grand majority of lawn mushrooms are harmless and, other than an unsightly spot in your yard, are doing absolutely no damage. They're breaking down organic matter and they're a sign of healthy, biologically active soil. So before you bring in a backhoe and remove all the dirt from your yard, maybe consider if you might actually be able to live with these interesting and ephemeral toadstools. If you decide that they must go, do not follow the Google advice to spray them with soap, salt, or baking soda. These may make the fruiting bodies decay a tiny bit quicker in theory. They usually last less than a week anyway. But the damage you will be doing to your lawn, to nearby plants, and any bodies of water that may receive the runoff far outweighs any damage that it's doing to the mushroom. It also does nothing to affect the underground mycelium, that is the actual organism the fruiting body is being produced by. If you want the mushrooms gone, pick them, toss them into the woods or your compost pile. Gloves aren't necessary. As we've covered, all fungi are safe to touch unless you're off put by maybe touching a slug hiding under there. Application of liquid copper and other commercially available antifungal solutions are also a wasted effort. They may help combat fungal turf diseases, but the amount and time of application required to eliminate and prevent all the fungi from your yard would be a ludicrous and expensive effort that wouldn't stop future fruitings. Trying to eliminate all the fungi in your soil would be like trying to eliminate all the bacteria on your body. You could bathe in bleach, I guess, but should you? So what about the last one? Surely removing all the soil from your yard would work, but putting down new soil will eventually be repopulated by fungi. So what do you replace it with? We already established several fruiting fungi like growing on wood chips, so that's out. You could do a rock garden or pave it with asphalt or concrete, I guess. Or can you? These are real photos of mushrooms pushing their way through concrete, rock, and asphalt. And it's not a terribly rare occurrence. So no, in short, besides hand removing individual fruiting bodies as they pop up, your time is better spent appreciating these amazing contributors to our ecosystem than trying to eliminate them. So that wraps up my presentation. Does anybody have any questions?
Oh, you have a wonderful sense of humor.
Thanks. I have to poke some fun when you get asked the same things pretty regularly. I have to, you know, I actually found I found lawn care companies, one namely in the UK, that offer a patented fungi removal program that you pay for, that is them bringing in a backhoe and removing all the soil from your yard. And it blew my mind. I'm like, why? Okay. Jamie, if you want, we have a Facebook group, Chattanooga Mushroom, Chattanooga Mushroom, something like that. If you find us and add, if we start putting something together, that's where we'll be advertising it. Mushrooms of North Carolina by Bassett et al. Also an excellent book. I highly recommend anything by the Bassetts. They're also friends of mine, but they're very knowledgeable and their books are well researched and comprehensive. Thank you, Terry.
I have a question. It's Hill Craddock again. For full disclosure, I'm back from Italy.
When you said that earlier, I was like, that has to be, there can't be that many hills.
That's me. No.
Excellent. Let's chat, shall we?
Okay, but
I'll send you an email.
Okay, my question is about the fungarium, the herbarium in Western Alabama. Can you tell us a little bit about what that is and what does the curator do?
Okay. I first started collecting fungi. It started because we got a grant through the Fungal Diversity Survey to sequence they were going to pay for us to sequence mushrooms, but we had to deposit them somewhere where they could be studied by future mycologists, and they would be available for research, and they would essentially be there in perpetuity. Should something come back novel and it need to be described, we would have type specimens available. And so we got in contact with Auburn, and Auburn had about 20 macro fungi in their fungarium. Everything else was micro fungi due to a previous curator, that being his focus. Or it may have even been the director. So I worked with them for a little while, and then I worked with Kevin England. And the problem with Auburn is once I deposited my specimens there, I couldn't do any more research on them. I couldn't do any more sequencing or anything. And so he put me in contact with Brian Keener over at West Alabama. So they really weren't doing fungi. They do lichens, but they weren't really keeping fungi. And I said, I have a team of collectors in Alabama. I have 20 some people that collect mushrooms from me. We send them to researchers all around the world. And he was just absolutely tickled to have me start. So I collect fungi. I have these mushrooms that are sent to me from other people who find them. We photograph them, we document them, we document where they were growing, what they were growing on, basically all the data that I was talking about in the beginning of the presentation. And we save them. They're saved in like a museum so that future researchers can come and study them. We can potentially describe new species from those collections, and basically they're available for future research for future mycologists. And we have specimens in herbaria across the United States that have been there for 200 years that we are still using to trying to use to solve mysteries. So much like a plant herbarium, it's the same concept.
Thank you. That was a great answer. So you're the assistant curator?
I'm the assistant curator, but I'm the only one who touches any of the mushrooms. Yeah, I dry label, catalog in mycoportal, and then send them down there for them to be packeted.
So you said they're stored in like a museum. The specimens, are they in paper pouches or boxes? I'm curious about some of the details. I'm sorry if I'm boring the rest of the people, but these are important questions that I have for my own research collections.
So I'm teaching them how to handle the mushrooms. They are lichenologists and plant people. So those are packeted. That's very easy. But they didn't have any freezing protocol. They were bringing in mushrooms that were full of insects, things like that could be potentially devastating. So we've established a freezing protocol that everything gets frozen. First, I freeze it in a deep freezer here before I even process it. And then once it goes to the herbarium, it gets frozen for 48 some hours again, just to make sure that we're not bringing in anything. And then those may need to get dehydrated again or put in a drying cabinet again, briefly, depending on the moisture level of the freezer. And then depending on the fungi, some of them get boxed, some of them I send in tiny little more structured containers, basically. Some of them are just in envelopes.
Okay, yeah.
I'll come up one day and we can dive into
just
some
curious,
details.
like a round number, how many accessions do you think are in the herbarium at West Alabama of fungi?
I think we've just crossed over 1,000, but I have another 2,000 here that I am processing.
At home. Very cool. Very cool. Thank you.
You're welcome.
A number of people are asking for your email.
Sure. I will just drop it in the chat. And this is, I normally give out my Alabama Mushroom Society email, but I'm going to not be president at the end of the year and won't have access to that. So this is my mushroom email. mycologymaven@gmail.com. You can send me emails there.
Do you
Do
find
you find
anyone?
anyone attempting to grow wild mushrooms? There are lots of people that there. So within the mushroom community, there's sort of a couple different niches. There are the people that are just really into identifying mushrooms. There are the people that are into the hardcore science, which is me. And then there are people who just want to find mushrooms to eat. And then there are the growers. So there are lots of people that are just into cultivation, and I can tell you almost nothing about cultivation. I can give you some very broad information. I can give you contacts to find locally produced grow kits that you can just kind of water and put on your counter and grow oyster mushrooms right in your kitchen. But if you are trying to establish a chestnut bed and are asking details, I'm not your girl.
Another question about how to do a spore print
Sure.
for
Okay,
ID.
so firstly, spore prints have a very limited use as far as ID. There are very few cases that a spore print color is going to be a defining characteristic. Somehow, somewhere along the way, that has gotten an awful lot of weight put on it. But if you're looking at the macromorphology of the fungus, usually you don't need a spore print except in very few cases. However, it is a lot of fun and they do make really cool art. So if you cut the mushroom off, so you just have the cap, you want to remove the stem, and you're going to set it on a piece of paper or a piece of tinfoil. A lot of times, if the mushroom's really wet, it's going to make your paper get wet and gooey too. So if you are able to sort of suspend it on toothpicks or something, so it's a little bit above it, and then you want to cover it with a Tupperware. You want to create a humid environment in there that is going to stimulate that mushroom to drop its spores, and leave it overnight. And then in the morning, you're going to take up that container and lift up the cap, and you're going to have a brilliant spore print, which, I mean, they can be purple, pink. One of the common lawn mushrooms makes a sickly green spore print that's just beautiful. And some fungi are easier to spore print than others. You're looking for the fungi that have gills usually. But it can be a fun little experiment. Kids love to do it. Does anybody else have any questions? Comments?
Well, I just have one. I think I posted it over to Karen. This isn't mushroom related, but I'm with the Tennessee Naturalist Program, and we use these classes for CEUs. So I'm just wondering, how can I prove that I was here?
And put your email address in, and I'll send an email confirming you were present.
Okay.
Okay.
Because every hour counts, you know.
Oh, yeah.
All right, thank you.
Uh,
I have another question, if I may.
Go for it.
It's Hillcraddock again.
Absolutely. Go ahead.
You told us you don't like the auricularia.
They're not one of my
They
favorites.
have
No.
I think they taste like rubber bands.
Yeah. Well,
But
the
I
texture reminds me of that chicken gristle that you get in the end of your
But
chicken.
I do like hot and sour soup. So in that context, they're essential. It seems like you mentioned a few, but if you had to pick like a favorite group of locally abundant edibles, what would be your favorite? I'm just curious.
Well, I don't know about locally abundant, but and it's actually one that I didn't mention, but it's Ericium coralloides. So it's the crown tooth coral, and it's my favorite. Most edible mushrooms I'll leave in the woods. And that one I will lug home every single time, and I make mock crab cakes with it. And I also really like lobsters. They tend to grow in pine dominant areas, or that's where I have found them, and they're worth the time and effort for me.
Okay.
But yeah, most mushrooms I'm like, I'd much rather find a teeny tiny, really interesting ascomycete than a oyster mushroom any day.
Okay, very cool. Thank you very much.
Uh-huh.
Parisium coralloides.
Yes, I'll type it in the chat here. It's the coral tooth. It's similar to the Ericium that my spelling might be
You showed
slightly
a
off.
picture of, yeah, Erinaceus. Yeah, very cool. Very good. Thank you.
Well, hey, Alicia, this has been fabulous. Lots
so
of
glad you enjoyed
interesting
it.
information and oh, just so much.
Yeah, it's a lot.
I'm
It's a lot.
intrigued. I have the rust galls down on my red cedars, so I'll go down and collect them now. Get rid of them because I do have a couple crab apples too, so that could be part of the issue there.
Well, chances are your apple trees are already infected. If they're not new and just popping up, removing your rust galls isn't going to help. Not your trees, unfortunately.
Well,
They're
maybe somebody else's then.
yeah, they're pervasive little things. They're fascinating, but they are brutal.
How do you get rid of them? I mean, other than removing
Yeah,
them, just put
yeah,
them in the trash?
burn them. You have to burn them. I mean, if you put them in the trash, you're just moving them from one place to the other, but put them in the burn pile.
All right. All right. And sounds good. This has been absolutely delightful. Excuse me. Maybe you would want to do a joint hike with us
Sure,
with
absolutely.
TNPS at some time. We're always looking for new and intriguing places to go on a hike, and having a focus on the fungi along the trail might be an interesting addition to a normal hike. So,
Oh yeah, I'd love to.
okay,
Hiking and looking for mushrooms is what I do.
yeah. As long as you wouldn't mind all these tag-alongs.
Not at all. No, I love leading hikes. I used to lead a monthly 4A before I left Alabama. That's
Uh-huh.
a great time. I lead a lot of homeschool groups, and yeah,
Oh
I've
yeah.
led lots of hikes. No, it's fantastic. We sometimes don't get very far. If it's a good mushroom year, we might only make it 10 feet, but we can hike.
Sounds like some of
Carrie,
our
send
botanizing
me an
hikes.
email. We'll exchange some contact information.
Our botanizing trips are, we have 18 field trips, 19 field trips listed this year. And we have two different speeds. People who are hikers sometimes come along with us and they're always disappointed because
They're like, you
we
guys
don't
really
go at
aren't doing
hike
much hiking
speed.
on this hike.
We do, we botanize.
Yep, yep.
So
Plant
we have
people and
hiking
bug
speed
people
and
are my
botanizing
speed.
speed.
Yep.
We have to have time for some people to lay down on the ground and look up at the undersides of the plant and take a good picture.
Absolutely. This sounds like my kind of hike.
Well, you get along grand with all of our folks. Oh, thank you so much. And it's been a delight to meet you. And I'm sure we can collaborate on some things along the way. And I'll be happy
Yeah,
to get this
absolutely.
posted on our webpage for seminars so that those that didn't make it tonight, because maybe they're on vacation, can come back and enjoy your talk. It's also
All right.
the kind of talk that some people are going to want to watch it 2 and 3 times.
There's a lot. I
You
tend to
packed a
info dump a
lot in
little
it.
bit, but take
Yeah.
good notes.
Yes. Well, with the video, it'll make it really easy for folks.
Absolutely.
So to pass along what some are writing in the chat. Thank you, Alicia. Thank you very much. Thanks for the presentation. Oh, I sent you a friend request. I work at Scotts Gulf Wilderness State Park. So let
Oh,
me know
yeah,
if you ever want to hike and look for mushrooms. Okay.
absolutely. I'll look for that.
That's Kat. I don't know who her Kat's last name's. Hike sounds so fun. Yes, to hike after we get some more rain. That sounds great. Ann Valentine. All right. And Ann Paget, I will send you an email shortly. Okay. So thank you everyone for coming. That said, I'm going to say good night. And thank you again, Alisha.
You're
This
very
has
welcome. Thank you
been
for having me.
wonderful. We appreciate it. We appreciate your time. Thank you.
Have a
Good
good
night,
night.
everyone, and let's pray for rain.